Is that full screen? Is that a full screen presentation, Maria?
Yes, yes.
Great. Okay. Yeah, so I was really excited when Maria got in contact with me and Ian, the other first author on this paper that we put out last year, looking at the global ocean size spectrum from bacteria to whales. So it's nice to speak to people who are in the size spectrum space. Yeah, hopefully if you've got questions on the methods or the theory or whatever, hopefully I can answer them and you find this interesting. But I thought to start, so first of all, just a bit of an overview of what are we talking about. So I think I'll start taking a step back and thinking about using size spectrum theory as a lens to assess human impacts. And then we'll look at a bit of the history of the size spectrum and specifically the Sheldon hypothesis. So there was size spectrum theory before Sheldon, but it was Sheldon's paper, I think, that back in the 70s, it sort of kicked off the last 50 years of size spectrum observations and size spectrum modeling. So we'll talk about that. And then we'll finish, well, those 1, 3, 4. And then we'll finish with looking at using the global size spectrum to assess humanity's role as marine apex predator. So can we do that, or how we did it? So let's take a step back. We know that we're living in an era of unprecedented change. So on the one hand, we have climate impacts are affecting marine ecosystems at the global scale. And so that's through things like in warming waters, changing distributions of primary production, declining primary production, ocean acidification, deoxygenation, and all those things are putting pressure on marine ecosystems and the biomass that they support. At the same time, you not only have a changing climate or a changing environment, you also have humans who are extracting more from putting large pressure from fish extraction, so extracting biomass from the oceans as well. So we sort of have these two pressures, both coming from humans, but in different ways. And so, as I said earlier, changing gears now to say modern size spectrum theory really was kicked off with this paper in 1972 by Sheldon et al. And they put this hypothesis out there. So they looked at the frequency distributions of particles between 1 and 100 microns. So that's 3 orders of magnitude or 2 orders of magnitude, sorry, on length, but 6 in terms of weight. And they said that to a first approximation, so this was only looking at plankton, but they sort of took a step back and said, well, okay, we're going to put out a hypothesis that to a first approximation, based on what we've seen in our samples, that there's a roughly equal concentration of material at all particle sizes from bacteria to whales. And so what that means, I mean, people in this group, we're fairly familiar with size spectrum modeling, but if we look at, if we disregard all the biodiversity in the oceans and just look at the size of organisms, and if we're counting up all the organisms in an area, a region, or on the global scale, weighing them, and then we distribute them into logarithmically equal size bins. So from 1 to 10 grams, 10 grams to 100 grams, 100 grams to a kilo, that there's a roughly equal concentration in each of those bins from bacteria to whales. And so this is a figure from their paper. So you can see there is a bit of a change in particle concentration on the y-axis, but this is that's in the context of over 6 orders of magnitude in body size change. So yeah, we're looking at a roughly equal distribution. And so this theory has kicked off about the last 50 years of size spectrum studies. And as I mentioned before, living in an era of unprecedented change, these sorts of studies, so people have used size spectrum theory to make observations as a metric of marine ecosystem structure, so actually going out and counting systems. And so this is from a study looking at the size spectrum literature from 2012 to 2017. So people focus on different parts of the size spectrum, but then in terms of counting things, but then also in terms of applications, we can use the size spectrum theory to build models to look at the impacts of fishing, climate change, interactions between fishing and climate change, diversity, habitat loss, and a lot more. So size spectrum theory has been well used to look at impacts of human impacts on marine ecosystems and to try and understand the structure of marine ecosystems. But from what we could see, me and my co-authors, was that Sheldon's hypothesis say at the global scale, so if we were able to count and weigh everything in the oceans and put it on a size spectrum, which was sort of in the spirit of Sheldon's original hypothesis, does it hold? So as far as we could see, that was still an open question in the literature if we tried to get a global spectrum. So just want to acknowledge my collaborators. So we came together to think about this question. So Ian and I led this paper together. So Ian is at the Max Planck Institute in Leipzig in Germany. So he'll be giving a similar talk for the second spectra group later. And then Yinon Bar-On from Weizmann Israel, who if you've seen his paper in PNAS, it's like the global biomass of life on Earth. So we thought, if we're going to do this, we should talk to someone like Yinon, who has experience with counting big numbers, and then also Eric Galbraith from McGill, who has a background in marine ecosystem and biogeochemical modeling. We also, though, we were in contact with a lot of other experts in the field. So, Anthony Richardson, Jason Everett, did a fair few others as well. So I wanted to acknowledge them. So we set out to build the global spectrum. So to do that, so first of all, just to set the boundaries on what we were doing, we decided that the Sheldon spectrum is looking at suspended particles in the water. So we considered all life from the surface down to the benthos. So we did talk a bit about, should we be including life in the sediments? But we decided not to do that because, Sheldon was counting things suspended in the water column. So we focused on that. So we had 5 major groups that covered the majority of life. And you can see that for each of these groups, there were different methods. So we used a combination of georeference samples. So for heterotrophic bacteria, there's the Pangea dataset, the Tara Ocean Survey, and there was a few others as well we used. For zooplankton, we used primarily the copepod database, which had over 200,000 georeference, so latitude, longitude, depth, and time samples. So for bacteria and zooplankton, we built statistical models to scale, to use, fit to those samples, and then chlorophyll, sea surface temperature, and depth. So pretty basic statistical models. For phytoplankton, we used another statistical model with satellite chlorophyll that gives you like the total biomass in the euphotic zone of phytoplankton in those 3 size classes. So pico, nano, micro phytoplankton, small, medium, and large. For fish, we used marine ecosystem models from FishMap. We chose, there's about 10 global marine ecosystem models that can give you fish biomass. We only chose 2 of those, and those had been constrained explicitly by fish catchers. So we thought, they could maybe give us a better estimate. And then with mammals, we used survey data and a few statistical approaches to estimate the biomass in mammals. And so putting that all together, what we did first, so there were 2 steps here. First of all, we needed to establish the size ranges of these groups. So the body mass ranges, smallest and largest sizes. So we did that just by looking through the literature at, what do people do in models? What does the literature say? And then we had the body size ranges. Then using those different approaches, we estimated the biomass of each of these groups in the global ocean and in the top 200 meters, so the epipelagic zone. So we looked and we considered both of those. We talked about both separately because there's different processes going. So they're both intimately linked. But we weren't sure as well, when Sheldon was talking about the size spectrum, it's not clear if they were talking about the top 200 meters or the whole ocean. So we decided to look at both. to distinguish them. Obviously, with an approach like this, there's huge uncertainties in what we're doing. So we try to capture as many sources of uncertainties in our estimates of our biomass, the different groups. So, for example, with bacteria, what I mean by a fold uncertainty. So we have the global ocean biomass is about 14 gigatons of wet weight biomass of bacteria. A 3.85, say a threefold uncertainty there means that, well, what we're saying is that it's sort of like a 95% confidence interval on a log space. What we're saying is that we're 95% confident that the global biomass of bacteria is between 5 and 45 gigatons. And so the median there is about 14. So you can see there's big uncertainty. So groups like macrozooplankton are the largest uncertainty. So it's over an order of magnitude that we have here. So putting that, so we have our size ranges and we have our the biomass of each size range. So now we're able to put it together into a size spectrum. So we distributed the biomass of each group within its size range. We looked first at the global abundance spectrum. So, we know if you want to go from a biomass spectrum to an abundance spectrum is just the number of organisms in a size range. And so if it's an equal distribution of biomass of small things and big things, well, you need more small things to make up that biomass. So yeah, you have a lot more small things and a lot less big things. But we did find that the slope of the abundance spectrum was quite close to -1, which if we go back to the biomass spectrum means that, yeah, we do have a spectrum that's fairly flat. But, we can see here that, so this figure, the solid areas is the upper 200 meters. So that's where we fit the size spectrum, is looking at the upper 200 meters. But you can see as well, those bars in each bin are the errors. And so there's a fair amount of error. There's big uncertainty as well. But what we concluded from this is that Sheldon et al. Were broadly right, except for bacteria and whales. So if we look over the entire ocean, there's a bit more bacteria, or even if we only look at the top 200 meters, a bit more bacteria and a lot less whales. And so we talk a bit in the paper about what might be, what is structuring the spectrum globally. There still are a lot of questions about that. But obviously, what I was talking about with uncertainty, so we wanted to make sure that that slope of about -1.04 for abundance or -0.04 for biomass, we wanted to make sure that that's how robust was that estimate to the assumptions we made. So first of all, because what we did when we were fitting our standard spectrum, if we had the size range of nanozooplankton, we would distribute the biomass of that group equally in the size bins in that range. And so what we thought about is, it could just become pretty circular, that if you're distributing the biomass in each one equally, well, it's not really a surprise that you get an equal distribution when you add it all up. So we tested, first of all, so the first column here is how does, doing, I think it was 10,000 samples or more, but what's the range, the distribution of our global biomass size spectrum slope if we assume we know nothing about how to distribute the biomass of each group. So that's just randomly distributing biomass in different bins within each group's size range. And so we found it was quite, it still a pretty narrow range. But then we don't only have that uncertainty, we also had huge uncertainties in the biomass of each group. So not only in how you distribute the biomass, but in how much biomass there actually is. So the second column is the range of slopes that we got when you vary not only how you distribute biomass for each group, but how much biomass there actually is. And from that, we were sampling from that 95% range for each group. And so we still found that at the global scale that it's still quite that given the magnitude or uncertainties, the range of our slopes was relatively small. So if anyone has done regional size spectrum studies, you know that even within a region where you've got a lot more temporal variability and that sort of stuff, you get a much bigger range of possible slopes for a single ecosystem. So at the global scale, we found it was pretty robust. So we had our size spectrum, and so and we found that Sheldon et al. They were broadly right, and so we thought now we could use this to assess human impacts with the global size spectrum. And so we were looking at two main human impacts, ones I briefly touched on at the beginning. We looked at fishing and we looked at climate change. So for fishing, we pulled together a synthesis of fish loss from stock assessments. So these are examples like worm and branch or ecosystem models. So the Christensen et al. And a few others as well. So that was to calculate, that was to get an estimate of fish loss. We also looked at published impacts of climate change on different taxa. So Kwiatkowski gives estimates on lower trophic levels from Earth system models, Lotzer et al. And Brady on higher trophic levels using marine ecosystem models. We were also though able to directly calculate climate impacts using, so as I said earlier, for bacteria and zooplankton, we had statistical models that we built. And then for fish, we had fishmet models that run climate change simulation. So we were also able to estimate climate impacts on bacteria and phytoplankton, zooplankton using our own statistical models. And we also looked at what it said in the literature. So pulling that together, we looked at the percentage loss of biomass comparing fishing to the present versus worse or business, it's not business as usual, high emissions climate scenarios. So RCP 8.5 between now and the end of the century. And so what we found is that at least on the upper 2/3 of the spectrum, the impacts at the global scale of part of historical fishing far exceed what is arguably a worst case projection of climate change between now and the end of the century. So again, big caveats, uncertainties, there's processes we're not including, but we thought that was surprising to us. It's maybe it's not surprising if you've been in the literature longer, but for us to see it visualized this way, that was quite sobering. And then also to see the impacts of climate change as well, sort of magnifying those things, especially on lower trophic levels. So when we visualize these impacts back on the size spectrum, here we have our pristine biomass spectrum. So we've taken out those error bars. This is looking at the top 200 meters and what humans have removed. So we've just scooped off a big chunk of the top 2/3 of the size spectrum in terms of biomass. And so we think we finish by thinking about this from an energetics perspective. So as far as we can tell, at a global scale, what's sustaining all the biomass in the oceans, the majority of the biomass in the oceans, is net primary production. So that's phytoplankton creating new organic matter that gets fed through the system and sustaining all that biomass. So as far as we know, compared to the magnitude of the loss in the upper trophic levels in terms of biomass and production, there hasn't been the change. There's a lot of uncertainty here as well, but the change in global net primary production is a lot smaller compared to the size of what we've removed. And so what that means is that the amount of energy lost is a lot bigger from the size spectrum compared to the energy gained in terms of humans and how much biomass we as a species create every year from fishing. So we did a few back of the envelope calculations to see that the amount of energy that was lost from what we've eaten is greater than 10 gigatons a year. The amount of energy, so that's what we've removed was producing about 10 gigatons of new biomass every year. But what we've gained in terms of new human biomass is 2 or 3 orders of magnitude smaller. And so this remains a really interesting, to me, I think this is a really interesting open question, is if we've removed the top third of the size spectrum globally, or we've taken a big chunk of it, where is that at? And so net primary production hasn't changed in a similar way. Where is that extra production going? And so as far as we can see, that's a pretty open question. So to finish, I spoke earlier about like an era of global change, using the size spectrum as a lens to look at global change. What we found is that Sheldon's hypothesis is broadly true at the global scale if we look at global biomass. But we also found then that human activities have already altered one of life's larger scale patterns, the global ocean size spectrum. We don't know the implications of this, except that they're, assuming other parts of the system haven't been as directly impacted, there'll be massive changes to energetics in the marine ecosystem. And then also that, the origin of this pattern, so understanding the mechanisms underpinning this pattern will really help to understand the impacts of these changes. So we need more work to actually understand what's driving the structure of the size spectrum and how we altered those mechanisms. All right, thank you.